now we stopped here and we talk about how, what are the different criterions and how we do molecular docking study using various computational tools. But with all these things, you know that there is some limitation in computational docking also. Although we have tremendous success, still there are some limitations. And the current challenges in docking are trade-off between efficiency and accuracy, and then more effective scoring functions and better model of flexibility. So those are the three limitations that we face, and we address all these problems by first we perform the docking study and then we perform large-scale molecular dynamics study so that this flexibility and accuracy, everything can be overcome, okay, to some extent. So docking, so generally the virtual screening, that is the first step that I have shown you, that HTTPS, that takes 10 seconds per molecule. And so that one is the fastest method, but it is the least accurate, that you can see from this, looking at this figure. And on the other hand, molecular docking is more accurate than the virtual screening, but molecular docking is also not that accurate. Okay. So now to overcome this, as I said, what do we need to do? So we use virtual screening and molecular docking for reducing the number of compounds from millions or billions to say 100 or say 20. Okay, suppose you reduce to that number. Now we want to further it down, and we want to also study what would be the impact of the ligand binding on the protein structure. Although, because that is the flexible nature of the protein, and I told you that after binding with the protein, with the ligand, protein can undergo many conformational changes. Okay, so we want to study that thing. For that one, we want to use simulation study, high performance computational study. So we use generally we use molecular dynamic simulation method, because we are also interested in studying the time evolution of the behavior of the ligand binding or the dynamics of the protein as a function of simulation time. Okay, Monte Carlo does not give you that kind of thing, that time is not there. Okay, so in MD we can do that thing in molecular dynamics study. So the thing is that in molecular dynamic simulation is very basic thing. Okay, you don't need to know much. What you need to know is the Newton's second law. Okay, so you know that from Newton's second law of physics that force is equal to mass time acceleration. Okay, and force is equal to negative gradient of potential energy, and acceleration, if you want to write it in terms of differential equation, it would be d2r by dt2. Okay, so then what you have basically, okay, so what you get actually is now, being a computer science student or computational guy, you may think that what is so difficulty in this thing? This is just second order differential equation, and you have to integrate it to get position, that is r, as a function of time. So that means you have to do the integration only. Okay, because we want to see how each atom of protein and ligand or the drug are moving as a function of simulation time. Okay, so that means you have to, this is the integration problem basically. This is a second order differential equation. But the problem is that this is a second order coupled differential equations, and generally this is very complex problem. So for example, the Bharat Biotech or this Oxford drug, this Covishield that they have designed, these vaccines, they are targeting the spike protein of the SARS-CoV-2 virus. Okay, for this COVID-19 I'm talking about. Today I'll talk only about COVID-19. Okay, why not show other examples? So now the spike protein is really big protein actually. You know, it's a trimeric protein. It's a huge, humongous protein, and some portion of it remains in the water and some portion remains in the membrane environment. And if you consider this water and membrane and the protein, everything in atomistic detail, then the total number of atoms would be 1 million. Okay. So if there are 1 millions of atoms, then there would be 3 millions of degrees of freedom. So if there is 3 degrees of freedom, it means you have 3 million this coupled second order differential equation, and you have to solve all of it simultaneously. Okay, so that's the problem. Okay, so it's a large scale simulation thing that you are simulating such a huge system. Okay, so now how do you do these things then for such a big system? And this potential V that we calculate, okay, basically we have to calculate force acting on each atom, Fi. I is the ith atom. Okay, so you have to just take the negative gradient of V. That means you have to differentiate the potential energy. Now the potential energy looks like this. Potential energy looks like this. It's a complex function. It has 5 different components. First one is the van der Waals interaction energy. Second one is the electrostatic interaction energy. Third one is the bond stretching energy, because bond can stretch like this, because we model that each atom is connected with another atom by spring. Okay, so that's why that allows the vibration of the atom. You know that at room temperature, temperature means energy. So at room temperature, your body is at room temperature, no, even if it is a little bit higher actually. So that means each atom is getting energy. And the energy is kBT, where kB is the Boltzmann constant and T is the absolute temperature. So if it is 300 Kelvin, if I say, at room temperature, your energy would be 0.56 kcal per mole. Okay, so if it has this much of energy, then each atom is vibrating. So that means there could be bond stretching. So that we are modeling that the bond does not stretch so much. There is no non-linearity. Okay, we say that we can model it using a harmonic potential. So that is Kb r minus r0 whole square. Okay, we are not considering any anharmonicity in the movement, but in principle there is. But if we include all these things, it will look very bad actually. So just simple, it's harmonic potential. Similarly, the angle can also be vibrating like that. Okay, so that is also we are considering that there is not large scale fluctuations, small scale fluctuations. Okay, so it can also be represented by a harmonic potential, and same thing is for the dihedral angle, the torsion angle, that is also harmonic potential. So these are the form of 5 different components of the total potential energy. Okay, so you have to make a differentiation of this function for each atom. Okay, then only you will get the force acting on each atom. Okay, so that's the thing. So then you plug in here. So then you will get. So in the third equation, basically, if you just put the value of V, then you will get that differential equations that we are going to solve. So now you can say that you can solve it using trapezoidal rule, or you can say that I will use RK4 method. So different methods you can use it, but those are very unstable. You cannot use it for these things. And when you have, as I said, that's a couple differential equation, and the number of equations increases drastically with the increasing number of degrees of freedom, and the number of degrees of freedom increases with the number of atoms. So if you have a bigger system, bigger protein, then the number of degrees of freedom increases drastically. And also the number of degrees of freedom comes from the water molecules or the membrane also, okay? Because everything we are representing it at the atomistic scale here. So this equation we can solve it using various methods, but the most popular is the Bartlett algorithm. He was a French mathematician. So what he says that first you take a third order Taylor expansion of RT plus delta t, where delta t is the time step. We are integrating it, okay, to get the time evolution of R. So the integration time step would be delta t. You just do that thing. So it would be RT plus R dot delta t, and R dot means dRdt, dRdt is V. Then the third term would be half R double dot into delta t whole square. Now R double dot means d2R dt2, that is acceleration. Okay, so these are the way then you can represent it. So this is for forward time. Okay, t plus delta t. You know your position at time t, and then you want to predict the position at time t plus delta t. Now you take a backward step. So you want to calculate RT minus delta t. Okay, again you do the Taylor's expansion, and then you will get something like that. Okay, now you add this RT plus delta t plus RT minus delta t. If you add these two together, what you get basically 2RT plus AT, A is the acceleration, delta t square plus O delta t. So that is the fourth order term, we will neglect it. So that is the error you are incorporating. Okay, that because we are deleting that part. Okay, so that's the error. So error is of the order of delta t to the power 4. And now if you rearrange these things, because you want to predict the time step in the future, okay, RT plus delta t. So then RT plus delta t would be 2RT minus RT minus delta t plus AT delta t whole square. It means that if you know the current position, that is the position of the atom at time t, and also you know the position of the atom at future time step, previous time steps, that is RT minus delta t. And if you can calculate and the potential you know, okay, so if you know the potential, then you can calculate the force, and if you calculate the force, then you can calculate the acceleration also. Just force divided by mass is the acceleration, so you can calculate the acceleration at time t. So if you have all these things, then you can predict the position of each atom in time step t plus delta t. Okay, so we'll just integrate like this way, and we want to study the position of the atom as a function of time. We'll evaluate it. So you have a landscape, rock landscape, and you want to see how your atoms are moving on that surface, and finally the objective is that it is moving in such a way that we will reach to the global minima. Okay, so that is the objective. Generally what happened, just let me show you one figure that shows how actually the structure actually, you know, it the protein structure. So this is this funnel you can see. So this is the landscape, free energy landscape. Okay, and you start from somewhere else, but your job is to reach to this end, the native state. That is where the correct three-dimensional structure is formed. And you can see that there are a lot of multiple minimas are there, and you may get stuck in one of those minima. You have to overcome that minima and reach to the native state. That is basically the job. Okay, so when you do MD simulations, basically all these positions of the atom that we are calculating in the future state, basically in this energy surface somewhere. Okay, And exhaustive search means you have to search this entire phase space. So that is the challenge. So so once you this RT plus delta T, you can calculate it, and then velocity also you can calculate it using this formula. Velocity, why do we need velocity? Velocity we need to calculate the kinetic energy. And why do we need kinetic energy? Kinetic energy is the manifestation of temperature. Okay, because if we are simulating a system at 300 Kelvin, because we have to simulate it at a biologically relevant temperature and physiological conditions. Okay, so now here you can see that in the case of V, you are introducing a second order error. On the other hand, for RT plus delta T, the position you are introducing a fourth order of error. Okay. Now there are a lot of variations of Valdez algorithm. There is a leapfrog algorithm where you seek to improve the velocity estimations, and then there is velocity Valdez algorithm also there. Okay, we will not talk about that. So just one such algorithm is good enough, that Valdez algorithm. So that's the algorithm. Okay. And first I will show you one test cases. One test cases is so most of the drugs currently which are under trial, okay, for, I'm not talking about vaccine, I'm talking about the drug, okay, because vaccine is already in the market. So now this drug in the left hand panel is, I have shown the SARS-CoV-2, that is COVID-19 3CL protease, okay. So this protease basically helps in the replication cycle, okay, of the virus. So now if you can inhibit its enzymatic function, then you can arrest the disease. So this is the target here. Now if you show in the left hand panel, you see that there is a small green surface I have shown. So that is the drug binding site. Now here I have considered two drugs, the B and C. The B one is a bulkier one, C one is a smaller one. And I told you, if you remember it, that your drug should not be too big. It should have molecular weight less than 500, but it should not be too small either. It should not be too small that it can freely rotate, move, translation, movement there inside the binding pocket. Then that will be a good candidate. Okay, so let us see whether that is hold true or not. We have one bulkier drug, one smaller drug candidate. Now, in this case, generally, to understand how the protein drug interacts, you can study it from the X-ray crystallographic, that is the experimental structure also. So you determine the crystal structure of the protein ligand complex, and then you visualize the structure and try to find the mechanism of binding. But then you are missing some aspects. What are those aspects? You are missing that effect of solvent. All biological macromolecules are residing in either in membrane or aqueous solvent. Okay, and water plays a major role in their structure actually, and also structure function relationship. So that part is missing. You cannot do it. And another thing, what is the role of the entropy in the binding? That also you cannot determine from the experimental study. Not only that, you cannot tell even from experimental. For example, you can do isothermal titration calorimetry study to estimate the binding energy between the protein and the drug, but you cannot tell what are the different components of the total binding energy. What is the contribution of idiostatic energy to the binding? What is the contribution of van der Waals interaction to the total binding affinity? So those kind of things you cannot calculate from experimental study. So that's why the job of molecular dynamic simulation or the computational study is to complement the experimental study. Whatever is lacking, we want to provide it and make a complete study. Okay. So now, if the experimental structure is not available for the protein ligand complex, you can generate it by virtual screening and molecular docking. So that's the thing, you can do it. Now there are various statistical methods that you can use to calculate the binding energy. One is the docking that we talk about, but docking is very fast method, it's the fastest method. but it is also the least accurate method. On the other hand, free energy perturbation or thermodynamic integration, that is the most accurate method, but it is very time consuming. So we make a compromise between speed and accuracy, and we choose MMPBSA method, where MM stands for molecular mechanics, PB stands for Poisson-Boltzmann, and SS stands for surface area. So this shows this is the linearized version of Poisson-Boltzmann equation. It's a partial difference. So Poisson-Boltzmann is a nonlinear partial differential equations, but nonlinear partial differential equation is very difficult to solve it computationally, numerically, because that's unstable. So you can convert it to a linearized version and you can solve it. So that's the thing. This method we are going to use it. It's very easy method. Okay. You start with the complex structure. That is, complex means the protein plus docked ligand or the drug molecule. Okay. And if it is not available, then you dock it. Otherwise, if the experimental structure is available, then you start from the experimental structure. And if it is a globular protein, then you have to put the entire thing in water box, okay, to mimic the biological environment. Because when you solve a crystal structure, you don't solve it in water or membrane environment actually. What do you do actually? You use a buffer solution and solve the crystal structure, and that is not the real biological environment. Okay, so you have to mimic the biological environment. You put it in a box of solvent, and if it is aqueous protein, so it is water. Then you run molecular dynamic simulations. So, and you store the conformations, that is only the XYZ coordinate, say every 2 picosecond time interval. The delta T that we use in MD simulation is 2 femtosecond. Why femtosecond? Because the fastest movement present in your system is the hydrogen bond, hydrogen atom. Hydrogen bond, they fluctuates a lot, very fast movement. Okay, so there are frequencies in that range. So you have to use it more than that. So that's why we use 2 femtosecond time steps. Okay, so now think about it. So for a bigger system, in this case, basically the system that I have shown you, we have around 70,000 atoms. 70,000 atoms means 2.1 degrees of freedom. So we have to solve 2.1 lakh couple differential equations. Okay. And to study these things, many of the processes occur, biological things occur at nanosecond to microsecond to millisecond time scale. Okay. So think about it. You are starting from femtosecond and you have to come to microsecond or nanosecond. Okay. So that means from 10 to the power minus 15 to 10 to the power minus 9, or if it is microsecond, then 10 to the power minus 52, you have to come to 10 to the power minus 6. So it means that you have to have iteration of 10 to the power 9 iteration. 10 to the power 9 iteration for, in this case, 2.1 lakh this differential equation. You have to solve it like that way. So that is the challenge. Okay, so that's why it is very difficult to perform it for a long time scale. So now you store these conformations, that is the XYZ.
The cherry on the head. Doesn't it look so cute? Also, it's an art exhibition, you know. Boring stuff. Nisha Chaudhary, nice profile picture.
Can you please mute yourself? Okay, thank you. So now by performing MD simulations, like you are coming from 10 to the power minus 15 to 10 to the power 9, okay. So you are storing energy, say each 2 picosecond or 10 picosecond time interval. Okay, so you will have an ensemble of conformations, structure, basically the XYZ coordinate with water molecules. So first, then you post-process it. So you remove the water molecules, then you will have an ensemble of protein, that is receptor plus ligand only. And from this, if you remove the ligand conformations, then you will get receptor configurations. And if you just remove the coordinates of the receptor, then you will have only the coordinates of ligand. So that means from these single simulations, you can have three different trajectories, one for the complex, one for the receptor, one for the ligand. Then you calculate the free energy, okay? And you use this formula. This is like GRL, that is the free energy of the complex minus free energy of the receptor minus free energy of the ligand. That will give you the binding free energy. That will give you the strength of the binding, how strong the ligand binds with the protein. Okay. Now, EG, that is the Gibbs free energy, that you can estimate it using the thermodynamic laws, that is G is equal to H minus Ts, where H is the enthalpy, T is the temperature, and S is the entropy. And in our case, enthalpy is nothing but sum of the molecular mechanical energy and solvation free energy. And entropy, that you have to estimate using normal mode analysis. Okay. So that is the thing. So generally, this is first box is the molecular mechanical energy and the solution energy, that you get it like this. And the entropy is the most difficult part. It's a time consuming thing, especially the vibrational entropy. Because I told you that when the drug binds to the protein, it loses its rotational, translational, and vibrational degrees of freedom. So you have to, so though that will basically give penalize, and that penalty is called the entropy. And you can calculate it using normal mode analysis. Okay, so normal mode analysis is basically it's another kind of machine learning algorithm. Okay, so you just calculate, you create a Hessian matrix, you just solve it, and then you will get the eigenvectors and eigenvalues. Okay, so then from there you will get this basically this age vibration, this formula, you put all this the frequency, the mode frequency, and then you will get the vibrational entropy. Okay, so that's how generally we calculate all these things. And this is very memory intensive calculations, the entropic part. You need really huge memory of your system if the, and computer systems, if your system increases rapidly. So the first case, then we show it for that two drug that I have shown you, and I have to test which one is a better drug. Okay, and I remember that I said that the drug molecule that you should design, you should snugly fit it into the binding pocket. Okay, if it is smaller than that, then it will move around the binding pocket, and that would be a very good drug candidate. So before that, so what do we actually run for 100 nanosecond time steps, total simulation length, and the time step that we use 2 femtosecond. Okay, and the simulation length is 100 nanosecond. Okay. So for first, before we do any kind of analysis, first we have to make sure that the simulation has converged. So one of the criterion for understanding the convergence of simulation is to evaluate how the structure is deviating from the starting structure, whether we have reached to the equilibrium or not. Okay. So one such measure is called the root mean square deviation. Okay. So the C alpha atoms of each amino acid, I study their deviation with respect to the starting conformation. Okay, and then that is plotted as a function of simulation time. That is the figure A. Okay, so you see that after 14 nanoseconds, you have reached to the equilibrium. There is a flat line basically. Okay, there are some fluctuations, but that's within the standard deviation. So you can say that the system has reached to the equilibrium. Now look at how about the, that is for the protein. Now how about the drug? For the drug, what do you see that? Look at the green one. Green one is the alpha ketoamide 13B. That is a bigger drug one. And the blue one corresponds to the smaller drug. So the bigger drug one, it fluctuates of course, and then it has a larger fluctuations than the blue one, that's the smaller one. But what you can see that if you just look at the frequency distribution, that is the probability distributions, then you see that the green one is characterized by a single peak. On the other hand, the blue one is characterized by two peaks. So that means at room temperature, this small molecule is very flexible. It can adopt two different conformations. And I told you that if you have a multiple conformations in the binding site, then that cannot be a good drug candidate. But alpha ketoamide, that shows that it adopts only a single conformation. So that is good actually. Okay. So this kind of thing you can study using experimental thing. You can use computational technique to study these things. So next thing we use one machine learning approach. So that is called K-means clustering algorithm that we use to find out the domain and conformations that is present for this ligand in the binding pocket, and that also agrees with these things. For B, you see that there is 64 and 36%. Those two different conformations are present. So that makes it a slightly less effective drug. So next thing we want to study how the motion of the atoms will change when the drug binds to the protein. So for that one, we use this thing called dynamic cross correlation map. Okay, so we calculated using these equations. So our delta R is the position. Okay, as a function of time, basically, you can just do these calculations. So now you plot it. Now you see that A is when the drug is not bound to the protein. So if you look at that position, so basically we make a color map, and this cross correlation, since this is a correlation, so it will vary from +1 to -1. +1 means highly correlated, and -1 means highly negatively correlated. Okay, that means highly anti-correlated motions, and 0 means no correlation. So now look at in figure A the R1 region. So you see a lot of patches are there, which is brown color patches. So you see there are a lot of positive correlations there. But now you see that when the drug binds, the same region, R1 region, if you look at B, what do you see? It has become more yellowish. Yellow is still positive correlation, but it is less correlative. So the correlation actually decreases. And look at the R2 region. So you see a lot of red color patches, which means there is a lot of anti-correlated motions. But in A, you see a lot of blue patches. That is also anti-correlated, but less anti-correlated. So in that region, the anti-correlated motion increases in R2 region. And in R1 region, the drug positive So now in C, that is for the small drug. You see that you got similar kind of movement. Also, you have noticed that in the R2 region for C, you see also a large scale anti-correlated motions. But what you see specifically the R3 region, you see some difference. In R3 region, in B, it is deep blue. That means it is anti-correlated motions. But in C, you also see there a lot of red patches. So that means in that region, the anti-correlated motion increases. So those are the different atomistic motions that you can you can predict what happens when the drug binds to your protein. Okay. So next thing, another way to understand the different dynamics, the different structural things. Another unsupervised machine learning algorithm that we use is called the Pcr algorithm, that is principal component analysis. Okay. Why do we need principal component analysis? Because we have to reduce the dimension of the data. In this case, the dimension of your data is 6n. Okay. Why 6n? You have 3 degrees of freedom that you get from the position XYZ. Okay, and then you have the kinetic energy part. Kinetic energy is momentum square divided by 2m. So momentum is px, py, pz, xyz coordinate. So 3 degrees of freedom that you get from the momentum and 3 you get from the position. So there are 6 degrees of freedom for each atom. So that means the dimension, overall dimension is 6n. Now I told you that for this one I have almost 7,000 atoms. So its dimension, the data that you will have, it will be 4.2 lakh. But can you visualize 4.2 lakh data? No, you cannot make any sense. You will say, sir, I will make a matrix. Well, I mean, you can make a matrix, but can you make sense any of that? You can make a table, still you cannot make a data. I mean, sense of it. So we perform principal component analysis, this machine learning thing. We want to see what are the dominant changes, okay, along certain directions, okay, without losing any information. So we want to reduce the dimension of the data without losing any further information. So for that one, we just compute this covariance matrix, okay, that you can calculate using this thing. Then this covariance matrix that you generate, you have to just diagonalize it. You can use Gauss elimination method or any linear algebra method that you have learned. You can use it to minimize it. But remember one thing here, it is a very, very big system. The dimension is pretty high. It increases with the increase in the number of atoms in your system. So for this kind of diagonalization, actually, you need very large memory of your system. So now, if you just diagonalize it, you will get the eigenvalues and eigenvectors. Okay, so eigenvectors are the direction in which you see the large-scale motion, and the eigenvalues determines the fluctuations in that region. So then, what you can do basically, you don't need to, and you will get a lot of principal components. Okay, but generally, first 2 components are the most important because where you will see the most fluctuations. So now you can see that the 6n dimension data, you can reduce it into 2-dimensional data. Okay, but remember one thing, that this principal component, PC1, PC2, they do not have the same attribute that you are using in the original data. It is something abstract concept. Only thing that you need to make sure you have to interpret your data correctly that you get from the principal component analysis. So we use this thing for these cases, okay, this principal component analysis. And generally in physics, we love to represent everything in terms of energy, it's free energy in terms of that. So we convert this PC1 and PC2 in terms of energy. How did you do it? We can just convert it, just multiply it minus RT and logarithm of this probability of these things, okay? The ratio of this, you just take it. Minus RT ln PC1 minus PC2. So that will convert it into an energy. That's called Boltzmann formula actually. So now what you see that when in the case of A, when the drug is not bound, you see two minima, 2 blue dot that you can see. And if you do then cluster, and then if you do the K clustering, then you will see that 67% corresponds to 1 minima and the other 33% corresponds to the other minima. So I have shown those 2 structures using blue and orange color. So that I have shown you. Okay. Now, when the bigger drug binds to the protein, you see that there is only 1 minima. So the structure is more stable. Okay, that's what you want. And C corresponds to when the small drug binds. And you see that again there are 2 conformations. It behaves like the apo structures, that is when there is no drug. Okay, so again for this one you do the K cluster analysis and you will find that these 2 minima, how much structures belongs to each minima. Okay, it's 61 versus 39% that. So those are, you can show it like that. Okay. So then automatically you can tell that the second drug, that is a small drug, is not a good candidate, because you are by structural characterization, you can see that this inside the binding pocket is very flexible. It is still not been able to stabilize the conformations of the protein. Okay. Now, extreme right, ABC, I have shown you this, the porcupine, this spike I have shown you. So this is called the porcupine plot. Okay. Just like in porcupine has a lot of spike on his body. It's just like that. Now here, the spike tells in which direction those atoms are moving. The fluctuation is there. Okay. And the length of the spike tells you the amount of fluctuations. Okay. So now looking at these things, you can see what is the impact of binding of the drug on the movement of the protein. So this figure you can relate with the dynamic cross-correlation map. You can relate it. Okay, this is another way of representing. You can see how different drug influences different kind of atomic motions in the protein. Okay, actually that also eventually determines why one drug is better than other drug for that particular protein. So these are the different analysis that you can perform, like different machine learning as well as different statistical and mechanical methods that you can use to understand this protein drug binding process, and that can also help you to further characterize and improve the lead optimization of your drug molecule to make it even a better drug. So that's one thing. This is also for the, I don't want to talk about it. This is for, this is actually the dihedral principal component. So initially that principal component that I have used, where I have used XYZ coordinate as the, for that XYZ coordinate, Cartesian coordinate. Now, sometimes in protein cases actually, as I told you that here the structure is determined by the phi psi angle. That is the dihedral angle or the torsional angle. So that is actually the internal coordinate. So you can do the same thing for the principal component analysis, but for the dihedral angle also you can do it. And generally, we use it for the loop region. Why for loop? Because loops are very flexible. It can adopt multiple conformations. Why do they do that? Because loops are generally exposed to the solvent. Okay, so they interact with the water molecule. So that's why they are very flexible. So now if you just implement this dihedral principal component analysis, then you can see that what are the different flexibility that a protein can adopt when it is free, when it is bound to a larger drug molecule, or it is bound to a smaller drug molecule. So you can see that for C, there are three different conformations. This small loop region they can adopt. This loop is very important actually. It connects these different regions. Okay. So these are the different implications of drug binding on the structural aspects of protein. Okay. So this is how we use large-scale simulation to study the dynamic nature of protein. Now, looking at this, now I told you that the small molecule, since it's more flexible in the binding, that is not a good drug candidate. So to ascertain it, let us calculate the binding energy. We use that one. So, and also told you that in experiment you cannot tell the various components of the binding free energy. Okay, so that's why we use simulation method. So here you see that for alpha ketoamide, that bigger molecule, the van der Waals interaction and the electrostatic interactions both are more favorable. It is more negative than the small molecule. Okay, that's why alpha ketoamide is a better drug candidate than the smaller one. Okay, because if you just calculate the total binding energy, delta G total, the estimated binding energy is like that get minus 9.05 kcal per. And for that the second drug molecule is minus 3.25 kcal. It means that the alpha ketoamide binds more strongly with the COVID-19 protease than the second drug. Okay? Now, if you remember and read newspaper, then you will know that initially when this COVID-19 break out, we didn't have any drug, and especially the doctors from Thailand say that they found that HIV, that is the AIDS drug, are effective. Okay, so lopinavir and darunavir. Can you please mute yourself? So lopinavir and darunavir are two such drugs which are basically AIDS drug. Okay, HIV drug. So now AIDS drug actually target the viral protease of HIV. Since these are protease drug, so then the doctors think, okay, why don't you use this protease drug for COVID-19 protease? Although they are different, but let us try and use it. And they say that they see some effect. So we wanted to study why did they see some effect. So we estimated the binding affinity with this COVID-19 viral protease, and we found that lopinavir and darunavir actually both are binding with a good binding affinity. Although it is not better than alpha ketoamide, but still it is good. So that's why initially when you read in the newspaper that they found that HIV drugs are working because of this reason, because actually it can bind with that COVID-19 viral protease. Okay, so you can answer a lot of these kind of questions. Okay, like doctors are trying different ways, but why they're working? Okay, what is the molecular origin? Why a drug works and why a drug does not work? You can tell that thing. Like this, the second drug Z31792168. Okay, so this drug, why it is not working? Because it binds with a very less binding energy. It has very less van der Waals energy, very less in electrostatic energy. Okay, and I also tell you that the polar formation and the entropy always disfavors the complex formation. That you can see that delta G pole is always positive for all cases, and also T delta S, that is also positive for all cases. That is the entropic term. So these are the two terms that always disfavors the complex formations. Okay, so you have to calculate those terms, because if you don't calculate those terms, then the estimated free energy would be more negative, and you think that okay, I have found a molecule which has very, very less binding energy. I mean negative energy. So that is the magic molecule. But once you calculate the entropy and polar solvation, then that will decrease significantly and it will become less negative. Okay, so that's why these are the two terms that you should always calculate to find the absolute binding free energy of the drug molecule with the protein. Okay, so next thing you can also calculate what are the amino acids those are contributing most to the binding free energy. Okay, because those are the hotspot residue that you can find it. Now, what is so it has two applications. One is you can find out which amino acids are not contributing more. So then you can optimize your drug further and make sure that it can interact with those amino acids and give a better binding free energy. One aspect that, and the second aspect is that by looking at this thing, you can tell which mutation will make sure that your drug will not work. Because virus also mutate, because already you know that there's a mutant strain in UK, and those strain actually died now spread in India also. Now scientists are telling that in Gujarat, already in Ahmedabad, they have that kind of strain before. That's why They have very fast. Even in Maharashtra also they have different kind of strain actually. Otherwise it will not. So, but one thing you should remember that if it becomes very infectious, okay, then it won't be that lethal. SARS-CoV-2 or COVID-19 is very infectious, and that's why the mortality rate is less than 2% in India. It's 1.44%. In India, there are other viruses, for example, Chandipura virus or KFD, Cyanosure forest disease virus, that is available in Karnataka, Cyanosure forest. Then I think you all have heard about this another virus from Nipah virus in Kerala. 2-3 years back, it got outbreak. Okay, so those are very lethal, but it's not infectious. If you are infected, only you are infected. You will die, but not others. There, in that case, in Chandipura virus, the mortality rate is 40%. Think about it. If SARS-CoV is that much lethal, okay, the 40% that out of 100, 40 people are dead, then what would be the case? So this is actually one thing good, that if it is if it is very contagious, then it will not be that much lethal. That's one thing good. But remember, Nipah can outbreak anytime. It is still in the avian. Okay, it is still in the avian means still it attacks the bat and other. But once it comes into the human, then God said, we have to be prepared for that. We have to start developing drug actually. Anyway, so this is one aspect. Basically, now looking at all these figures, you can optimize your drug. So these are the applications. Basically, the large-scale simulation. You can complement your molecular dynamics study using this kind of simulation to make a better drug candidate, and also you can understand why a drug works, why another drug is not working, why a mutation can cause a drug resistance. So those are the different molecular origin of this drug resistance, drug binding. You can study using large-scale dynamic simulations. I will show you one example, another. Okay, this is different thing I want to show you. Okay, another case I just want to show you is this one. Okay, now we want to study why SARS-CoV-2, that is COVID-19, is more infectious than previous one. So previous one was MERS-CoV, and then the first one was SARS-CoV. Okay, SARS-CoV outbreak in China in 2003, and MERS-CoV outbreak in Middle East in 2014-15, and now came the SARS-CoV-2, and we found that SARS-CoV-2 is more contagious. It is affecting globally. Okay, so there is no country which is not being affected with this disease. So the question is why? So for that one, we study this human one protein is there. It is called ISC15, and the PL-PRO, that is another enzyme present in the virus. Okay, polyprotease. So basically, it binds more strongly with the human ISC15 in case of SARS-CoV-2 compared to MERS-CoV on SARS-CoV. That's why actually it is more contagious. We want to study that thing, and we also want to study the dynamic aspect of it. So the green one is the ISC15. We want to study how the brown one binds to it. For this one, we study very large scale molecular dynamic simulations. Okay, here in this case, basically everything and the simulation length is 4 microseconds. So we run it in repeat simulation. So that means total 8 microsecond data. And we are using 2 femtosecond time steps. So you can imagine. So from 10 to the power minus 15 to 10 to the power minus 6, 10 to the power 9 step that we have. So actually 4 into 10 to the power 9 iterations we have done. Okay. So that is also in repeat. So the first thing again, we want to study the how flexible is this ISG15 molecule. Okay, so that one we have studied it. A1 is for SARS-CoV, B for MERS-CoV, and C for SARS-CoV-2. So you can see that because all peaks corresponds to different ensemble of conformations. So you can see that that when they are in complex, the structure actually is stabilized more. If you look at the figure C, okay, this APO, black one is APO, means when it is not bound to the protein. I mean, with the ISG15. In that case, you can see that it has 3 different, the black curve, if you just look at it, see 3 different peaks. But when it is bound, then the peaks have been shifted towards lesser. So that means the binding of this with the viral polypore is actually stabilize the structure. Second thing, we look at the radius of gyration. Radius of gyration tells about how compact the structure is. So this is the ISG15. So you can see that again, SARS-CoV, MERS-CoV, and SARS-CoV-2. All these cases that you can see that how the structure, the compactness is changing. Okay, with the binding of the other protein, it basically becomes expand. So the radius of gyration increases. But the main thing that we want to actually study, particularly this hinge angle. Okay, so you see these both two domains are connected by a small loop. So we just calculate the angle. We take the center of mass of the loop, center of mass of this upper lobe, and the center of mass of the lower lobe. We connect these 3 points using a vector. Okay, so angle is what? You just take a dot product, then you will get that angle. So you just calculate that angle, and then you can see that when this human ISG15 is not bound to viral polyprotease, it is in closed state. It means that its angle is very less, around 70 degree. But when it is bound, then the angle actually shifted, so it becomes more expand. So it means that the structure is elastic. I'm sorry, plastic. Okay, it has a plasticity. And especially if you just look at this thing, for example, we are interested in SARS-CoV-2, so that is actually yellow color. Okay, so in the case of SARS-CoV-2, the yellow color, you see that angle is around 100 degree. Okay, so it's pretty extended. On the other hand, for the blue one, that is for SARS-CoV, no, that is for MERS-CoV mutant. It has 2 different minima. One around 125 and another one at 110. 2 difference. Okay. So it tells that basically at room temperature how many conformations it can adopt. So this is the flexible nature of that protein that you can study using molecular dynamic study. And finally, we are interested to know why SARS-CoV-2 is more infectious than SARS-CoV and MERS-CoV. So for that one, we just estimate the binding energy between the SARS-CoV-2 polyprotease, that is PL-PRO, with the human ISG15. Okay. So now just look at the green box only. You can see that for SARS-CoV-2, the binding energy is much much higher than the SARS-CoV and MERS-CoV. So that means PL protease binds more strongly with the human ISC15 compared to MERS-CoV and SARS-CoV. And this causes de-isolation in human. Then we become very susceptible to the disease actually. Okay. So this way you understand now why SARS-CoV-2 is more infectious and more lethal than MERS-CoV and SARS-CoV previous to persons. Okay, so these are the different computational techniques that you can understand various aspects of disease as well as the host pathogen interactions. That means how you are getting infected, because if you understand the molecular origin of how you are getting infected, then that will help you to design a drug that can basically destroy that interaction mechanism. Okay, so these are the different applications of high performance computing in drug designing as well as to understand the protein dynamics. Okay, another example that I want to give you about this protein. Okay, computational investigation of structural dynamics of SARS-CoV-2 methyl transferase stimulatory factor heterodimer, this bound to the cofactor sum. Okay, so now I have shown you one example. First, I have shown you the 3CL proteins. So most of the drugs are targeting that. Okay, targeting that protein. Then I discussed with you how computer simulation can help you to understand why SARS-CoV-2 is more infectious and lethal compared to SARS-CoV and MERS-CoV. And final thing, I'll talk about another protein of SARS-CoV-2 that can also be targeted to design a drug. Okay. So I don't want to talk about the biology part because I'm not a biologist either. So this protein actually, NSP16, NSP10, this is a heterodimer that is forming. Okay, so it is known that NSP10 acts as a stimulatory factor to execute the methyl activity of NSP16 and is a common mechanism among coronaviruses. Okay, so basically same thing you will see for SARS-CoV and MERS-CoV, not only for SARS-CoV-2. So if you understand the mechanism in SARS-CoV-2, the same thing will be applicable to SARS-CoV and MERS-CoV. Okay, and not only that, the binding of NSP10 with NSP16 increases the binding pocket protein and extend the RNA binding group of NSP16. So we want to study its dynamics. For that one, again, we perform molecular dynamic simulations of, basically in this case, we performed a 10 microsecond simulation. Okay. So you can imagine how many iterations we have to do. We started from 2 femtosecond and then we went up to 10 microsecond. Okay, so 10 into 10 to the power minus 6, that means 10 to the power minus 5. So overall 10 to the power 10 iterations we have performed for this thing and for such a big system actually. So it also shows different, I don't want to talk about this, these are not that important thing. Mainly I'm gonna talk about the dynamic aspects, okay, this machine learning thing. So we have the trajectory, then we use this machine learning thing, this principal component analysis to understand the dynamics of it. So look at figure A, that shows basically when this NSP16, NSP10 heterodimer. Okay, so in that case, you have a single minima. If you just look at it, the dark blue is just 1. And then the second one is relatively, it is also minima, but it's not the global minima. It is relatively higher energy, but the energy difference is not that big. It is less than 0.5 kcal per mole. If it is less than 0.5 kcal per mole, and I told you that at room temperature each atom has energy of 0.56 kcal per mole. So that means that state is also accessible. Okay. So then you can also perform the K-means clustering, and then you can characterize which is which. Okay, I mean the global minima corresponds to how much of the total conformations. Okay, that percentage wise you can just calculate it. So it is 63 versus 37. And then you study this NSP10, that is for NSP16 when it is bound with NSP10. Then you look at the dynamics of NSP10 when it is bound with NSP16. So now you see that NSP10 is a little bit more flexible. It has 3 different minimas, but none are global minima. Okay, so it is constantly in dynamic equilibrium in among these three conformations. Okay, and then figure C corresponds to NSP16 monomer. So that is when it is not bound with the NSP10. Then also again you see that it has very narrow free energy barrier. Okay, although it behaves like same like A, but it is narrow. The conformational space is very stricter. And for NSP10 monomer, then again you see that the sampling space again further become very narrower. And there are 3 different minimas, and the corresponding K-means clustering are also shown. So overall, you can see that how the binding of another protein stabilize or destabilize the effect. Okay, so that's the thing. And then we want to just study the dynamics of the loop region. Okay, so it has different these loop regions. That's this long loop it has. We want to study its dynamics, and then you see that very interesting thing when NSP16 is in complex with NSP10, then the loop is very stabilized. Initially there is deviation. The red curve, just look at it. With time it is increasing, and then it started decreasing, and finally it reaches to an equilibrium and stabilizes at 1 angstrom. On the other hand, when NSP16 is not bound with NSP10, then initially it increases, the green one, and then stabilize at a higher value, around 3 angstrom. So it means that the loop becomes stabilized after the binding with NSP10. So that's why NSP10 acts as a stimulatory. Okay. So as I said that as a physicist, you always love everything to convert into energy. So the same thing, we just convert this into in the figure B, basically we have converted into energy. Okay, potential of mean force, because it is easier to explain everything in terms of energy. Now for dimer, you can see that I told you that eventually it got stabilized at a single value, lower value, 1 angstrom. So when you convert it into energy, you see exactly the same thing, that around 1.4 angstrom you see the lowest energy. But at the same time, you see 2 other minimas. Those minimas are located at less than 1 kcal per mole. On the other hand, for green, you see only 2 minima, one at 3.3 angstrom, and then the second one is a higher energy. Okay, that one is observed around 0.5 angstrom. But the energetic difference between the global minima and the second minima is almost 3 kcal per mole. Okay. So it means that the second state is not accessible. So only accessible state is this 3.3 angstrom. So it means that the protein, that is NSP16, is more flexible when it is in apose state. That means when it is not bound to NSP10. Now we want to study the binding of the SAM. Okay, so this small molecule we want to study, because most of the drugs people are, when they are trying to develop, because they think that since the drug is binding to NSP16, so I should not include NSP10. All this data I am showing you just to tell you, while you are developing drug targeting NSP16, you should include NSP10 also. That is the main study of these things. Okay. So here we show that basically this is again free energy, this SAM. So when just specifically look at this figure A first. So you see that when you consider NSP16, NSP10, this dimer, the binding is characterized by a single minima. On the other hand, when you have considered only NSP16 while you are developing the drug, okay, you are not considering the NSP10. Then you see that initially you started with the lower energy, strong binding, but when you run the simulation, gradually your drug molecule comes out of the binding pocket and you reach to a position which corresponds to the weak binding. That has the lowest energetic barrier. Okay, and the corresponding figure is shown in C and D with the position of the drug molecule in the binding pocket. So if we just look at D, you can see that I have shown you the initial position, and after running AMD simulation, the final, you see that you can see that final position is also shown. So it has moved away. So it also justifies that why do we need to consider the dimer while you designing the drug. So although a lot of paper have been published, okay, showing, I mean, they target only the NSP16, but later on we showed that, so this paper is published in Frontiers in Biosciences and Biomolecular Modeling. So then later, basically last year, around November, December, this paper is published. So then we showed that that's not the correct approach. And we showed that why it is not correct. What is the molecular origin? Why we should consider the dimer, not the monomer. Okay. So then again, we estimated the difference, various binding energy components, and then I showed you basically for the dimer, the binding energy is minus 6.8 kcal per mole. But if you consider the monomer, NSP16, the energy is minus 0.6 kcal per mole. So basically, it is not binding. No, I showed you that it is coming out. So it means that, again, it shows that why you should consider the dimer, not the monomer. Another aspect that that we should talk about, again, what are the different amino acids those are contributing to the total binding energy. Okay. Now again, you can see that when you consider, look at figure A, when you consider the NSP16 and NSP10 dimer, then you see that many amino acids are contributing to the total binding. On the other hand, if you consider only the NSP16 while you were designing the drug, then only a few amino acids are contributing to the binding energy. So that's why there is this difference in the binding strength. And that again underscores the need to use the dimeric structure to design the drug rather than the monomeric form. Okay. So these are all different things, and then I showed what are the different amino acids that interacts with interactions between NSP16 and NSP10. So here the green dotted line shows the hydrogen bonding between NSP16 and NSP10. On the other end, this red dotted line shows the hydrophobic interactions. Okay, so those are different interactions that you can see. What stabilizes these protein-protein complexes? I told you that when the NSP10 binds with the NSP16, then it increases the stability of NSP16. Why? Because it forms like this many interactions. That's why the complex structure is more stable than the monomeric structure. Okay, and this is the time evolution of the eyes to show the effect of temperature. Okay, so if you want to study like everything we have studied at room temperature 300 Kelvin, now say we want to study whether what would be the effect when if we elevate the temperature. Okay, body temperature basically. So then you can see that actually at 300 Kelvin the drug is strongly bound. Okay, but at 300 Kelvin, what did you see? After some time, the drug got detached. Initially, it was here. Then look at the second one, that it comes out of the binding pocket. But after certain time, again it comes back. But still, it is not bound at the exact position. Rather, it is bound at the surface of the protein. Okay. So it means that at high temperature, the SAM molecule disassociates from the binding site. So that is to show the effect of temperature. Now, what is the effect of salt concentration? Okay, because in our body we don't have the pure water. No, it's ionic in nature. We have a lot of salt in our solution. So what is the effect of salt? So generally at physiological salt is the 0.15 molar. Okay, so we studied it at elevated 0.25 molar also, and then you can see that in both cases we don't see. The generally 0.1 molar is physiological. Okay, we have increases it to 0.15 and 0.25. In both cases, you can see the COM means the center of mass distance of the ligand and the binding site. If it increases, it means the ligand is dissociating from the binding site. So you can see that there is a sharp increase in the distance. Initially, we started at 14 angstrom, then very short time, it reaches to a distance of 18 angstroms. So it moves 4 angstrom away from the binding site. Okay, so that means there is a slight destabilization of the binding if you increase the salt concentration. Okay, so you have to be very careful while you're designing all these things. Okay, temperature has an effect. Also, the salt also has an effect on the binding of these things. Okay, so with all these things, I will stop, and all these things have been done by my research group members. Okay, Nisa, Pulbabu, Rajeshri, Sayan. So they are the PhD scholar. They are working on it. And then also I would like to acknowledge DBT and DST for their support, and also MHRD for seed grant that I received. So thank you all for listening, and I'm ready to take any questions if you have.